The first step in cell respiration is glycolysis. The process harnesses energy through breaking a single glucose molecule into two pyruvate molecules. The process occurs in the cytoplasm of the cell. The initial step of glycolysis is termed energy requiring, where the energy is produced when two ATP molecules are used. The first ATP attaches a phosphate group to one end of the glucose and creates a glucose 6-phosphate. The glucose 6-phosphate is isomerized into fructose 6-phosphate. The second ATP attaches a phosphate group to the other end of the newly formed fructose 6-phosphate. The backbone of the molecule splits and forms G3P and DHAP. DHAP is immediately converted into G3P. Per G3P, a single hydrogen and 2 electrons are given to NAD+ to reduce to form NADH. At the same time, G3P combines to form 1,3-BPG, an unstable molecule, with an inorganic phosphate. 1,3-BPG releases a phosphate group to ADP, forming ATP. The end result of this stage is a molecule of 3PG, resulting in 2 ATP molecules forming from the G3P in a process called substrate-level phosphorylation. The 3PG molecule isomerizes into 2PG by rearranging its structure. 2PG releases 1 proton and 1 hydroxide ion, which form water, and leaves a molecule of PEP. Unstable PEP breaks down, releasing enough energy to transfer 1 phosphate to ADP, resulting in another ATP, or a net total of 2 ATP, 1 per PEP. The original molecule is now broken into 2 pyruvate with a 3-carbon backbone. Therefore, the end result is 2 NADH, 2 ATP, net, 2 pyruvate, and water as waste.
Pyruvate oxidation is the second stage in cell respiration. Membrane carriers help move the pyruvate molecules from the outer membranes into the inner membranes and finally into the matrix. Coenzyme A attaches to the acetyl group on the pyruvate and is then oxidized by NAD+ to create NADH. A carboxyl group is removed as waste in the form of CO2, carbon dioxide. The resulting product is acetyl-CoA, and energy is needed in the body when acetyl-CoA enters the Krebs cycle to continue aerobic cell respiration.
The third step in cell respiration is a Krebs cycle. It consists of 8 reactions from organic molecules into ATP, NADH, and FADH2, which is usable for the cell. For every glucose that enters glycolysis, 2 cycles of Krebs occur in the matrix of mitochondria. The Krebs cycle begins with acetyl-CoA from pyruvate oxidation, reacting with oxaloacetate to form citrate. The CoA is then removed. The citrate isomerizes into isocitrate. Isocitrate goes through a redox reaction and oxidizes to become alpha-ketoglutarate, and an NAD+ ion from the matrix is reduced, gaining electrons to form energy coenzyme NADH. During this step, CO2 is released as waste. Alpha-ketoglutarate forms succinyl-CoA through binding with CoA as well as being oxidized by NAD+, which reduces into NADH during the removal of CO2. Succinyl-CoA releases lots of energy while losing its CoA to become succinate. A phosphate located in the matrix displaces the CoA, GDP phosphorylates to become GTP. The phosphate is transferred to ADP, creating ATP. FAD from the matrix oxidizes succinate, forming coenzyme FADH2, resulting in succinate becoming fumarate. H2O is added to fumarate, forming malate. Another redox reaction with NAD+ turns malate into NADH, an oxaloacetate, and the cycle is ready to begin again. Therefore, in summary, the Krebs cycle produces 6 NADH, 2 ATP, 2 FADH2 after 2 cycles. At the end of the cycle, C and O are released as waste as CO2, whereas the remaining H+ ions are carried by NADH and FADH2 to electron transport chain.
The electron transfer chain. Mitochondrial membrane. The purpose of electron transfer chain is to create ATP oxidative phosphorylation. There are 7 major components of electron transfer chain. The first one is complex 1, which is NADH dehydrogenase. Complex 2 is the succinate dehydrogenase. Complex 3 is the cytochrome BC1 complex. And complex 4 is the cytochrome oxidase. There's also oxygen, which all of these extra hydrogens attached to, which we'll talk about later. And there's a big one and cytochrome C. There are many steps in the ETC, so bear with me here. The first step is the proteins. Well, these proteins here, here, are all in increasing order of electronegativity. NADH binds to NADH hydrogenase, which is the first component, and FADH2 binds to the succinate dehydrogenase, which is the second component. And while this is happening, H plus ions are pumped into the intermembrane space, which is this. UQ carries electrons from NADH dehydrogenase to cytochrome BC1 complex. Site B carries electrons from cytochrome B1 to cytochrome oxidase. FADH2 works the same, but starts at the succinate dehydrogenase complex. FADH also makes less ATP because it travels through fewer electrons. Zoom in. Our net production is NADH makes 2A, nope, makes 3 ATP and 3 hydrogen ions. Meanwhile, FADH2 makes 2 ATP and 2 hydrogen ions. 2 hydrogen ions and an oxygen found in the inner mitochondrial membrane bond together to make good old H2O. The reason why oxygen is used as the bonding, is because it's strong enough to strip the electrons from the membrane. The overall goal of electron transport chain is to create enough energy from electrochemical gradient to power the ATP synthase.
Chemiosmosis. Chemiosmosis only occurs after the electrochemical gradient is created. H plus ions now travel through ATP synthase in the membrane. H plus ions flow into the matrix. The flow of H plus provides energy to bond ADP and PI. This is how ATP is made through oxidative phosphorylation.